Okay, it will be 9. I think we can start. Your colleague, she just left again. Cannot see her. She was here, but she gone again. I hope she come back. Okay. Good. So we had a look at RNA yesterday, and I told you we will talk a bit about regulation. We talk about the promoter already in the eukaryote, and we said the polymerase, the RNA polymerase, has to bind there at the promoter site, and we talk about the cis and trans, where we said cis elements are, we can say, the sequence, the base sequence in the promoter. If that has mutations, then the gene behind it, the transcription or the product also may not be produced. Okay, because the mutation in the promoter sequence may cause a problem then for proteins that help the RNA polymerase to bind. Whereas if the transcription factors, proteins that bind to the promoter, if they carry mutations, they are called trans factors because they come from somewhere else and have to bind to the promoter. Now the promoter is the basic regulatory element of the gene. Every gene needs a promoter, otherwise it cannot be expressed, we cannot get transcripts. But it's just a bare minimum, okay? Normally a gene will have other regulatory elements. And for the eukaryote, you may come across two major elements that are called enhancer and silencer. Enhancer or silencer. Silen. So enhancer, as the name says, enhances the transcription rate. So you get more transcripts, whereas the silencer will suppress the transcription. These are also cis elements. That means base sequences that are in some distance to the promoter. Okay. So for example, you may have an enhancer somewhere upstream of the promoter. It may increase the transcription rate. It may also help to make the transcription tissue specific. So that in one tissue, the gene is actively transcribed, in another tissue type not. Okay. The interesting thing about enhancers and silencers is that you can move them. For different genes, it was found enhancers may be upstream of the promoter or downstream of the promoter. And the distance can be very large. It may not be just 10, 20 base pairs. It may be 100 of base pairs. It may be several thousands of base pairs away. Researchers even make experiments where they moved the enhancer, the sequence from upstream to downstream of the gene and brought it back into the organism. And bacteria or some frog oocyte, some simple eukaryotic organism like yeast also possible, and had a look what will happen, and the enhancer was still working. So the enhancer is a cis element to which proteins will bind, trans factors again, transcription factors. The question is if we can move the enhancer also in case of the silencer, which will suppress transcription. If we can move them around and they still work, how is that possible? How can something that is actually far away from the promoter have an effect on the transcription rate and affect somehow the binding of the RNA polymerase there and stimulate or suppress it? So for example, just for example, you could also switch the positions. So how it's again because the DNA can fold. What you think when you think about linear sequence that is far away may in reality be next to each other because the DNA just folds and E and P or P and S may be just sitting next to each other because the DNA is bent and is dynamic. It may change up to the situation in the cell. Okay, what's going on? So actually these elements sit close to each other in one or the other situation, and then the proteins that bind to these two different cis elements then sit also close to each other and have an effect on each other. Okay, and can either stimulate the RNA polymerase to bind and enhance transcription or suppress the ability of the RNA polymerase to bind at the promoter and lower the transcription rate that way. So these are possibilities that are used in the eukaryotic cell to regulate transcription. So we have many abilities to regulate gene expression in the cell. And so together with our intron-exon model that we have in the eukaryote, we are very flexible. Okay, for each gene, very flexible. We can have different transcripts from one gene, meaning different proteins from one gene, and we can regulate the transcription rate up and down with different elements, and we can also put some element there that allows it to be tissue specific in some way. Okay, in one tissue you get one transcript, in another tissue you get another transcript, in another tissue there is no transcript at all, it's blocked, and so on. Okay, so very flexible this way. How about the bacteria? Bacteria also have genes, much smaller number than the eukaryote, and now the bacteria, of course, also have a promoter. We could see that yesterday already. They have a promoter. The sequence is different from the eukaryotic promoters, but basically we also get transcription following after the promoter at some way. And if you read a microbiology books, maybe older ones, they may talk about cistron when they refer to genes. This is an older name. The microbiologists, they called a gene a cistron. And the genes in the prokaryote are a bit different organized. And it can be that behind one promoter, you actually have coding sequences for several proteins. They would call that a polycistronic gene now, because it's more than one protein product that you get. And so that would be, each would be a structural gene. And you have some regulating sequences close to the promoter that is called an operator. So yeah, we called it O for operator. To which transcription factors, we could call them bind, and stimulate or suppress transcription. So this unit of promoter, operator, and these structural genes is called an operon. Operon. So that is something that is unique for the bacteria. Okay, this kind of thing doesn't exist in the eukaryote. Now, that means you get one transcript. If you get transcription, and in the transcript, you have now three coding areas independently from each other that all have their own start and stop codon. The AUG and the UAA or whatever they have. So for each one, so you have three times start and stop codon for these coding regions, and you will get translation then also independently for these three regions. It's not like one ribosome has to move along that transcript and translate one after the other. No, it can be three ribosomes can be sit on that transcript and independently synthesize each protein. That is not possible in the eukaryote because we produce the transcript in the nucleus, and then we have to transport it out into the cytoplasm, and there we get translation. The bacteria don't have that. They have no nucleus. So they can do in parallel transcription and translation. The transcript does not have to be finished yet. They can start already when some part of the transcript has been synthesized and there is enough space for the ribosome to bind, then it can bind already and start. Okay. So operons, why? Why have bacteria this kind of model? This operon model, it was developed by some researchers called Giacomo. They said that this kind of unit exists in the bacteria before molecular biology allowed us to have a look at the sequence actually, just from their observations. What they noticed was that the enzymes are produced depending what is available in the environment of the bacteria. So bacteria will always try to be very efficient and only produce the enzymes that are required in the present situation and that allows them to be most energy efficient. The model that we have in the textbook for the operon will always be the lex at operon, like C operon. So that is the textbook example. You have 3 structural genes and that operon has proteins coded that are necessary if you want to metabolize lactose. The bacteria will not produce these proteins, will not transcribe this operon if there is no lactose in the environment, because it doesn't, you would say it doesn't make sense. So, but the bacteria don't think like that. Okay, they just have regulatory mechanisms where lactose actually is some kind of inducer that would stimulate transcription. So normally we have a repressor, a protein sitting at the operator, a repressor. And this repressor, as the name says, would block transcription. If lactose comes into the environment, then lactose can interact with the repressor, act as an inducer. It would change the structure of the repressor by binding to it, and then the repressor cannot bind so tightly any longer at the operator. So the chance for transcription goes up. Then these structural genes are used. That means transcription happens and proteins are produced, and these proteins are necessary to metabolize the lactose. That means the lactose concentration over time will go down in the environment. And if it's low enough, then not enough lactose there to act as the inducer, to bind to the repressor. That means the repressor then will change its conformation again and bind tightly to the operator again and block transcription that way. So this inhibition and stimulation stuff in biology, that is never 100%. You have to think there's always a small amount that is passing through. So you don't block 100% that thing, and also not stimulate 100%. If you would do that, the system would collapse. All these pathways allow a little bit to work, even under inhibition condition. So that way, the bacteria can work very efficient. So they produce these enzymes only when necessary. And that is also the case for other things. If you had, for example, different sugars in your growth medium, if you have glucose as well as lactose, then this one here would also not be switched on, even you have lactose, because glucose provides more energy. It needs less energy input to metabolize glucose, and you get more back. So it's more efficient first than to degrade the glucose and build your ATP or whatever you need, than to metabolize lactose. So if both sugars are there, then first the glucose will be used. And when the glucose is gone, then this one will be switched on and the lactose will be metabolized. Okay, let's have a look at some pictures. I have to go out of that one here. And operon. So, operon. Okay, so we have our promoter operator region and structural genes behind it. And if we have no repressor sitting there, then the genes will be on by default. Okay, and only if the repressor binds, then we switch it off. And normally the bacteria, they will have a repressor, so that things are switched off that are not required. They don't produce things just to produce them. They really do it only when they need them. So in positive control, you need to have some factor that comes and binds to the operator region and promotes the transcription. Okay. That is often the case in the eukaryote. Whereas the prokaryote, they like to have a negative control mostly. Okay, but not always, as we will see then. Here's that like C, the lag operon. You can see we have the three structural genes: the beta-galactosidase, permease, and transacetylase, and then in front, which is actually not in front, but far away on the circular genome of the bacteria, okay, that like like inhibitor that produced here, the like I gene that is producing the inhibitor, the repressor, and it's actually far away, okay? It's just not really sitting in front of the lag operon. It's just for the textbook, okay, now. So the repressor then normally when it's produced, it would go to the operator of the lag operon and sit there and prevent transcription here of this area, so that we don't get these proteins produced here, okay? So repressor and RNA polymerase bind at site that overlap around the start point of the lag operon. So you can see the promoter region and the operator region, they are somewhat overlapping, okay? And so the RNA polymerase has to bind at the promoter, and it can do that and unwind the DNA if at the operator no repressor is sitting, okay? When you observe what will happen in the cell, what kind of product amounts you will find, then when you act at the inducer, the repressor is a protein, but it is coded by a gene here, the LAG-I gene that has the code to produce the repressor. The repressor is the protein, the LAG-I is the gene, okay? So if we add the inducer in case of the lactose, okay, then after some time, the amount of LAG-mRNA goes up. Okay, because when we act the inducer, it will bind to the repressor. The repressor will go away from the operator, and so transcription can start. So there is a little bit, you can see a time lag, and then we can measure larger amounts of our RNA, and then it will go down again when we remove the inducer. And the amount of protein, the beta-galactosidase that is produced is a little bit later, because first we need the transcript, and after we have the transcript, then we get the translation a short time later, and then we get the protein. Okay, so but you can see this is the scale is just minutes, so there is just a few minutes between. Okay, we don't have to wait long. The repressor actually in this case is a tetramer. So there are 4 repressors that form a tetramer, and that bind at the operator region. Okay. When our inducer comes, so the lactose, and binds to it, then it's changed the conformation, okay, of the repressor, and the repressor cannot bind at the operator any longer. Actually, we have to say it can bind less good. It still can, but less good, much less good than without the inducer bound to it. Okay. So if we get mutations, for example, in the operator, then it might be that the mutation, the base sequence in the operator, if it is changed, that the repressor cannot bind any longer because of that change, which would mean that our transcription is going on and going on all the time. So now the bacteria would also produce these proteins even though lactose is there. If you have this mutant bacteria together with the wild type bacteria in the growth medium, then in the absence of lactose, the mutated bacteria would be at a disadvantage because they spend energy for enzymes that they actually cannot use. Whereas the wild type can downregulate that and have an advantage. They save energy. So they would outgrow the mutant bacteria in that situation. Only if you have always lactose there, then the mutated bacteria would have the same advantage as the wild type. Okay, so the mutation in that operator region is a cis mutation. Like always, it's in the place, it's at the DNA level. We can have also mutations in the repressor gene, which will cause a non-functional repressor. The effect would be the same. It cannot bind because the structure doesn't fit any longer, and you have production of these enzymes all the time. Okay, but this now, the mutation is in the trans factor, is in the protein. Actually, the mutation is somewhere in the gene for this that produce this repressor. Okay. So what I told you, this repressor binding or non-binding is not absolute. Okay, it's not 0 or 100%, but it's highly specific, let's say. So the binding constant here at the operator sequence is much higher, 2 times 10 to the power of 13, compared to other DNA sequences. There it will bind only 2 times 10 to the power of 6. So you can see it's a factor 7. So it means 10 million times, 10 million times better that it binds to the specific operator sequence than it will bind to other sequences on the DNA. So it's highly specific, but it's not absolute, okay? If the inducer comes, you can see the specificity goes down, and the affinity for this binding site. You can see by a factor 1, 2, 3 difference, so 1,000-fold down the specificity here, and still to the other DNA is the same. Okay, so it can still bind there, but less good. Okay, so you have to think in these ways. That is how biological systems work. Most of the time, it's not absolute. It's just allows you to regulate your pathways up and down in some way, but it will never be completely switched off. Okay. Good. So in the normal case, all the repressor will be bound somewhere at the DNA, at the specific site, and also if there is more than there are specific sites, then the repressor will bind unspecifically somewhere else on the DNA. Okay. Then if the inducer is coming in, then it's highly likely that it will also go away from the specific site and bind somewhere unspecifically on the DNA. Okay, and if you remove then the inducer, it will just move back to the specific site, because the specificity for that specific site, as we said, is much higher. Okay, so it's a dynamic process, changing all the time. That is what you have to think about the cell anyway. It's not just a picture that is static. There is something going on all the time up to the situation. Okay, here's another operon, this time tryptophan in the middle. And here they have 2 other operons, and they say the repressor that also binds to the tryptophan operon. It can also bind to other operator regions of other operands. Okay, so the sequences are a little bit different between them. So specificity of binding may be a little bit different between the different operator regions of the different operons. Now, tryptophan is an amino acid. Okay, the lactose was the sugar. So normally lactose, we want to degrade it to get energy. Whereas for the tryptophan, actually we want to synthesize it because we need it to build proteins. Okay, so it's a different thing. Operators can have different positions. They are not fixed. So if you look at different genes, you will find that your operator in respect to the promoter region that is here in red, this area, then the operator you can see can be in different places, okay, up to the gene. For each gene is in a specific place, but that place may be different between different genes, okay? So there's also flexibility, the same as the enhancer and silencer in the eukaryote. Here in the prokaryote, these regulatory elements can have also different positions. Okay. So we have negative control and we have positive control. Okay, positive control means we have actually a protein that binds and acts as an activator. Whereas in negative control, on the left side, we have something that acts as a repressor. Okay, so you can remember that if there's a repressor involved, it's negative. If you have the activator, then it's positive. Okay, they may have a co-activator or co-repressor, or okay, that bind to the repressor, change the structure, the same as for the activator that they can bind to and also change the structure, and in this case enhance the binding of the activator, whereas for the repressor, when it binds, it lowers the specificity to bind there. Okay, so now we have a case of positive regulation in the bacteria that works via this molecule. This is cyclic AMP. So we know normally if we have the adenine bound with the phosphate group, that we have a connection normally only at the 5' end. Okay, that is where our phosphate groups are attached to the base. Okay, but in this cyclic AMP, there is a second connection at the 3'. Okay, so that's why I call it a cyclic AMP. This is important for a regulation mechanism. I told you normally the bacteria, if you give them different sugars, they will first try to degrade the glucose because it needs the smallest amount of energy input to get the largest amount of energy back. Okay. So if you have glucose in the medium, then you will have normally a small amount of CAMP because you will produce a lot of ATP. Okay. This will cause a protein called CAP, also called catabolite activator protein, to be inactive and not to bind to DNA at the promoter operator region, and you don't stimulate transcription. Whereas if you have no glucose, then it means the levels of cyclic AMP will go up because no ATP is produced. Okay. Then this cyclic AMP will bind to the cap and then can bind to the DNA and stimulate transcription. This is a positive regulation mechanism. They have sequences where they bind to. You can see it's quite short. Actually, only 5 base pairs are required so that it can bind to. There can be differences in this sequence up to how much the sequence is changed. This cap protein can bind more or less good. Okay, if it's highly conserved, then they can bind very good. If you have some other base there, like what they indicate with the N, any other base there, then it can still bind, but less good. Okay. And you can see different operons for galactose, lactose, arabinose. They all use this kind also of positive regulation. They are also negative regulate. They have also a repressor to bind somewhere else at the operator, but they also have these cap binding sites. Okay, that are located in different places regarding to promoter and operator. Okay, so it means that these operons are negative and positive regulated. So it allows us, if you have two sugars, like glucose on any of these other series here present at the same time, to still prefer, if glucose is there, to still prefer the glucose, and because the CAMP level is low, then the positive regulation, no transcription, no enhancing there. And so the glucose is preferred. If the glucose is gone and the CAMP goes up, then you get the positive regulation and you enhance the transcription there. Okay. So you can have a mix, positive and negative regulation there. And that allows us, if you have more than one nutrient, to make a choice for the bacteria. Beside the lac operon, in textbooks, they normally give you also the tryptophan operon as an example of regulation in bacteria. Now, I told you the tryptophan operon is necessary if you want to produce enzymes required to produce the amino acid tryptophan. Now we want to synthesize something, the amino acid tryptophan. Okay, we don't want to degrade something. The interesting thing is that in that control region where we have the promoter and our operator, we have two more elements that are important in the regulation, what they call leader, which is coding for a so-called leader peptide. Okay, so a small number of amino acids that form this so-called leader peptide. And behind this leader region, they have what they call attenuator. The attenuator is a region where you can have stem loops that act as terminators. Okay. One thing that you should be aware of, that in that leader peptide, there are two codons next to each other for tryptophan. Tryptophan. Okay. Now, tryptophan, the amino acid itself, has an effect on the transcription of this operon. Lactose, in the case of the lac operon, stimulated transcription. But tryptophan makes the opposite thing. Tryptophan lowers the transcription rate. It's understandable because lactose, we want to degrade it. So we want to have the enzymes for it. But tryptophan, if tryptophan is present in the cell, then actually is no need to produce more of it. So it would mean only if the level of tryptophan gets lower and lower in the cell, then the bacterium will start to transcribe this tryptophan operon and produce the enzymes required to synthesize more tryptophan. So how does it work? You have to remember again that in the bacteria, transcription and translation happen in parallel. Okay? So transcription of the leader region can bind at the promoter, and it may pause here in that leader region, and that may be different how it pauses the RNA polymerase, depending whether no tryptophan or tryptophan present. No tryptophan, then the polymerase will move on and translation will start of the structural genes. If tryptophan is present, then we get this termination hairpin forms in the attenuator region, and the polymerase will, the RNA polymerase will terminate, and we don't get the proteins. So why the difference? Why, if no tryptophan is present, we get it, the translation, and if tryptophan is present, we get these hairpin loops. How does that work? What are these hairpin loops? So in that attenuator region, we have actually 1, 2, 3, 4 DNA sequences that are complementary to each other. So 1 to 2, 2 to 3, 3 to 4. So they can form different stem loops. So we could have 1 and 2, and we could have 3 and 4. 3 and 4 make the terminator hairpin if they come together. If we can get the regions 2 and 3 to pair, then this terminator region here 4 is single-stranded and cannot act as the terminator. So we need to get this one here to terminate transcription and to block production of the proteins. Okay. So why do we get in one situation that terminator hairpin, and the other situation we don't get it? In the presence of tryptophan, we get the terminator hairpin. If no tryptophan is there, or very little tryptophan, then we get this one, and we don't get the termination, and we get our proteins to produce more tryptophan. How does it work? Works like this. Tryptophan absent, the ribosome will stop at that place, because in the leader peptide, as I told you, there are 2 codons for tryptophan. If no tryptophan is there, then it means there is no synthesis possible, no translation possible, because without tryptophan, we cannot create that leader peptide. That means the ribosome stops at this point. Okay. and that will keep this kind of loop, hairpin loop, intact. Okay, and we cannot get the terminator hairpin. That means our RNA polymerase can continue to make the transcript. Whereas if tryptophan is present, then it means the ribosome can quickly move on, can pass this tryptophan because tryptophan is there. It can form that signal peptide without a problem and can move on along the transcript. And when it does that, then it interrupts this stem loop. Okay, it interrupts it. It cannot build, but then the 3 and 4 can base pair and make that terminator hairpin. Which will stimulate the RNA polymerase to stop further synthesis of RNA. This mechanism can only work in the bacteria because they do transcription and translation in parallel, in the same place at the same time. It's not possible in the eukaryote. The transcription and translation is separated. Attenuation, they call that attenuation. Okay, and it makes sense, this mechanism, if you want to synthesize something. In this case, the amino acid tryptophan. So you have two examples normally in the textbooks. The Lexed as the basic negative regulation and also with the cap protein positive regulation. And you have this tryptophan operon for the attenuation model there. Okay. So you can see bacteria have quite a number of tricks available, how they can regulate, yes, how they can regulate the transcription. Okay. Good. Then let's move on. From the transcription and let's go to the translation. The translation, when Watson and Crick developed their model of the DNA, then at that time, they start to think already, how is the information coded in the DNA? Because after DNA was found to be the genetic material, it has to contain the information. So where is the information? Then the only place where it could be is in the bases. And because there are only 4 bases, it cannot be a single base that codes for a single amino acid. Because what we want to do, the information for the proteins is we have 20 amino acids. 20 amino acids and we have 4 bases. So if you say one base codes for one amino acid, then you could have only 4 amino acids. So that would mean the researchers at the time thought already, okay, we need more than one base for the code. What happens if we take two bases? If we have two bases, that would mean like 4 to the power 2 squared. So that is 16. 16 possibilities, combinations of two bases. Okay, should not be enough because we have 20 amino acids. So they speculate at the time already, we need 3. 4 to the power of 3, 64, huh? 64. So that would mean if you take 3 bases, then we have 64 possibilities, 64 different combinations. That would mean we could actually code for more than 20 amino acids, but naturally there are just 20, okay? And so that would mean that either only 20 are used, or maybe there is more than one code possible for each amino acid. So we could have 3 or so for each, and then 4 left also. Okay, that was speculation at that time. Just you noticed you have this number of amino acids, you have this number of bases. Now let's think about it. How could the code look? The coding, what is it? So 4 bases, not enough. If only single bases are used, if 2 bases are used, not enough. If 3 bases are used, okay, it's more than we need. And but then you have to do experiments. You have to find out what the code actually is, because at that time, no sequencing. No one had ever sequenced any single gene at that time. Not possible. There was no technique available. Technique for sequencing became available in the 70s. So 50 years ago, Sanger sequencing became available. At that time, then the first DNA sequences became known. So how to do if you cannot look directly at it? Nothing. How to find out which combination of bases is the code for which amino acid? So you have to start very simple. So you take only one kind of base and you make short stretches of nucleotides that contain only a single base, like only A, or C, or G, or poly T. And you make an in vitro translation kind of system. And you look what you get as the peptide. Okay. And so it means you would now find out what is the code for 4 different amino acids this way. Because if you have 3 times A, or 3 times C, or 3 times G, or 3 times T, you get one specific amino acid in each case, and only that one. So that means then in the next step, you would have to use 2 bases, 2 different bases, and make stretches of nucleotides sequences containing only 2 bases. The problem was at that time, it's not like when you go nowadays and you want to do PCR, you just order a company and they make you exactly the sequence that you want to have. At that time, it was rather random. They had a way already to combine the nucleotides, but the result was not predictable. Okay, it was just combined. Now, if you combine two bases, like let's say A and C, you may get A, C, 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 or you might get AC, 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 or whatever. Okay, so it means you get several combinations, possible combinations already. When you have two, you can see that already when we use two or some that now it gets more complicated. Okay, so what they did, what the researchers did, they tried to use different ratios of the bases. So start to use a lot of A and very little C, for example. So the chance to get AC, AC, AC is very small in that case. So you have a higher chance to get A, and then occasionally a C. Okay, that way. And so they had a look how the proportion of amino acids that were put into the peptide is changing with the ratio of the bases that they put in their experiment. Okay, so it took many years to decipher the code, and in 1967 they had solved the problem. So in 1953 Watson and Crick published that paper of the structure, and they already said the code may be three bases. And in 1967, so 14 years, researchers worked on that problem to decipher the code, because they could not do it directly. No technique available. And so it took a long time to do that. In 1967, they had cracked it and had for all 64 possibilities actually found what's going on. So it's really that 64 possibilities are used. 61 to code amino acids and 3 to indicate a stop in the synthesis, stop signals. So all 64 possibilities are used. Okay. So we called it a base triplet because 3, or a codon. Codon is 3 bases, a base triplet that contains the information for one amino acid or a stop signal. So when you look at the genetic code, then you can notice that often the first two bases in the triplet are actually sufficient to let us know what amino acid it will be that is coded. So when, for example, you look here at the leucine, then you see the first two are always C and U. And the third one, doesn't matter which of the four bases it is, it will be always the same amino acid that is coded. You can see leucine actually has two more codons here with the UUA and UUG. So that one has 6 codons. If you look, the number of codons is different between different amino acids. There are 2 that have only a single one. Which ones? Which 2 have only a single codon? tryptophan and methionine. These two, you can see, have only a single codon. That means for these two, you need only, if you get a mutation in any of their bases, is something else. Okay? Whereas for the leucine, you can change the third base to whatever you like, it will still be leucine. So methionine and tryptophan are very critical. And normally we know when we look and compare in protein families the sequences, when we can see that methionines or tryptophans are conserved in the position in the family members, we know they have an important function for the protein. It might be if we knock that out by a mutation that the protein may not work any longer. Okay. Okay. Because of this fact that for many amino acids, if you know the first two bases in their codons, that we know what it will be, and that the third one is not so important then any longer. There was a hypothesis already. Is A or C begin with. Can you show it? What you want? UTT. Yes, so it is in the condition T or in such then I think it will be this now. Yeah, if you can change, then you will get something else. Another. Yes, you get another amino acid. Just putting to this sample. The protein will be still produced, but you don't know the function may not be okay any longer. No, because it's if it's highly conserved. No? Nowadays we have for many protein families, many members and can compare the sequences. If you see a tryptophan is highly conserved, then it means it's important. If, of course, in different family members at the same position there are different amino acids, then it would mean it's not so important. Okay, you can have different amino acids there, and the protein obviously different will still work. But if you have 10 family members and all 10 have tryptophan at the same position, then it means that tryptophan is really important. If you try to change it in something else, then it may not work any longer. The chance is high if that changes. Okay, so here when you try to summarize that, then you can see for the orange ones, for all these codons, the third base doesn't matter any longer because it will be anyway, regardless of the base in the third position, it will be the same amino acid. So 32 codons, half of the codons, it doesn't matter what is in the third position, it won't change the amino acid, okay? And the other colors, you can see here what it means. The purines differ from pyrimidines, that is 14 and 10 cases here. And then the one that are really unique is very small number only. Unique definitions. You can see here our methionine and the tryptophan. Okay. And here you have as well something. Okay. Good. Then it was proposed, I think by Crick already, that there is unusual base pairing allowed between the codon and the anticodon. Anticodon, you know where it is? It's on the tRNA, the transfer RNA. The transfer RNA has to bind with the anticodon to the codon of the messenger RNA. And it was proposed already that the third base may not be so important, which on the anticodon is the first base, because the complementary sequence, okay, they have to bind 5 prime, 3 prime, 3 prime, 5 prime, like in double-stranded DNA. Otherwise, they cannot make a double-stranded molecule. The RNA can also make double-strand, but also antiparallel, okay? So that means the third base in the third position in the codon would have to bind with the first base in the anticodon. So if you have A or G, they could bind with the U in the anticodon. G is specific, U is specific, but C or U with G again can make unusual pairing. The normal one would be GC, but also G and U can base pair in this case. The same as here, U and A would be the normal expected, but we can also get U and G. This will not create problems for the synthesized protein. Okay, it will be still correct. This is also called wobble. I don't know if you have heard that already. WOBBLE, wobble, base wobble. Here they have the word wobbling. Wobbling at the third position, base wobble. So you have some unusual base pairing allowed for certain combinations. You can see U with A or G with C or U, whereas the U, in this case, the G, is the important one. Another thing is that what I have shown you, the tables, is called the universal code. The universal code that we find, as the name says, in all organisms, but there are exceptions in the mitochondria. The mitochondria, as you can see here for different organisms, may have a slightly different genetic code where a few codons code different amino acid. Okay? In the mitochondria. In your nucleus of all eukaryote, we have the universal code. But if you look at the mitochondria, it might be slightly different. So that means if you work with a protein that is encoded on a mitochondrion, you have to be careful. If you sequence that gene and you use the universal code to tell the computer to translate it into the amino acids, you will get a wrong sequence. You have to instruct your computer software to use the codons that are used in the mitochondria of that organism to translate it into a protein sequence. Otherwise, you get a problem later on in your experiments. If you, for example, make a recombinant protein and you use then the universal code, but actually you should use the mitochondrial code, you have different amino acids in certain places in your protein and it may not work any longer. Or even you have a stop codon somewhere and you get no functional protein at all. So we have to know that. Let's have a look. E. coli, the different components. When you look at the different RNAs, the messenger RNA is the RNA important to make the proteins. You can see the amount of dry cell mass is only around 1%, different kind of molecules per cell and different types and copies of each type. It's actually very small because they produce only what they need at the time. But if you look at the tRNA and RNA, the ribosomal RNA, you can see the amount of dry mass is already much, much larger, and the number of the molecules is also much larger. Okay, copy number and so on. Also, ribosomal protein, 9% of the dry cell mass, a molecule per cell, 10 to the power 6 here, different types, 52, and copies and so on. So you can see, all these are required for translation. The translation process, when you add all this up here, is nearly a third of the dry cell mass of the bacterium. So it needs a lot of energy. Translation needs a lot of energy from the cell. That's why they like to keep or produce only the proteins that they really need, the bacteria, because it needs a lot of energy. If they would waste their energy to produce proteins that they don't need, they cannot survive. They have to be efficient. They are single-celled organism. They don't store big amounts. They live from what is there at the present time, okay, and have to be very efficient. So, and compared to our messenger RNA, we need large amounts of tRNA and ribosomal RNA, many, many more copies of them. Our tRNAs, normally when you see them as two-dimensional molecules in the textbook, they say they look like plants, small plants, like clover leaves, what they call them, clover leaves. So that is a tiny, tiny plants with, and that would be the leaf 1, 2, 3, and maybe you have a fourth leaf sometimes. Okay, so they form stem loops. Okay, these RNAs form stem loops like most RNAs would do in a natural situation. And you can see on this here at the bottom, there is the anticodon. So the three bases, number T34, 35, 36, they would form the anticodon that binds with the codon on the RNA, on the messenger RNA, okay? All tRNAs have the anticodon in this position. Their base sequence will be different, not identical, okay? And their structure is similar, but also not identical. And at the opposite side, what they call the acceptor arm, they all have the sequence CCA. And at the A, the amino acid is attached, because that is the function of the tRNAs. They are the so-called transfer RNAs. They transfer the amino acid to the ribosome. They bind via the anticodon to the codon of the messenger RNA, and then the amino acid that they carry will be connected via a peptide bond to the previous amino acid that the tRNA before, the previous tRNA, brought in to the ribosome. So that you get peptide bond by peptide bond, you're growing peptide, and at the end, your protein out of it. Okay, so that means this sequence is also very important. You can see invariant, this red, conserved. If you would have a mutation there, the A is not there, but something else, then it doesn't work any longer. Okay. The two-dimensional structure of this tRNAs actually goes into a three-dimensional structure that looks more like an L. And so you have the anticodon down here, and you have now here already added one amino acid to the acceptor arm at that CCA sequence. Okay. So how does that work? The amino acids and the tRNA are not binding directly by themselves. There are enzymes involved. These enzymes have names. They're called amino acyl tRNA synthetase. Amino acyl tRNA synthetase, and there are several of them because we have 61 different codons, 20 different amino acids. That means we also have a number, maybe up to 61 different tRNAs. And because everything works via structure, and because the sequences of the different tRNAs are different, and of course the structure of the 20 amino acids is also different, that means you need different of these enzymes so that the structure fits, because they have to recognize the proper tRNA, they have to recognize the proper amino acid and combine them. They have to attach the amino acid to the correct tRNA. So they have to be highly specific. Okay? Because after they did their job, after these enzymes did their job, there is no correction any longer. Whenever the loaded tRNA leaves this kind of enzyme, there is no correction possible any longer. Only as long as they sit at these enzymes can correction be done. If the wrong amino acid was added to the tRNA. Okay, as long as they still sit there, a correction is possible. Later on, not. Okay, so we need this. We said we need our messenger RNA, we need our tRNA. You could see we need the ribosomal RNA. We need the ribosomal proteins. So at the end, we need the ribosome. The ribosome is a mix of ribosomal RNA and ribosomal proteins. So gRNA, ribosomal RNAs are actually end products, and they are not never go to the protein level. They are produced as functional RNAs, okay, and are used as RNA molecules. And the ribosomal RNAs exist in combination with the proteins in the ribosome. And if you make mutations in their sequences, the ribosome will not function correctly in many cases. So they're important. They're also important for the function of the ribosomes, these ribosomal RNAs. And their base sequence is important. What else do we need? Ribosomes. Ribosomes are normally a small and a large unit, subunit. Okay. The ribosome size is a bit different between bacteria and eukaryote. In the prokaryote, they are a bit smaller. Okay, both the ribosomal RNA is smaller and also the number of proteins is smaller in the bacterial ribosomes, but the basic function is the same. Also in our mitochondria, we have our own ribosome because we can do translation there, and they are more like bacterial ribosomes in some ways than the ribosome in our cytoplasm. Okay. Ribosomes. So what else do we need? Energy. This process needs a lot of energy. And interestingly, it's often not ATP, but GTP or GDP that is used as the energy source. Okay. And we need translation factors. Translation factors. So the same as we have transcription factors that help in the transcription, we need now proteins that help in the translation process and are called translation factors. Okay, there are quite a number of them, and the number is larger in the eukaryote compared to the prokaryote. Okay, but we need them in both systems, and we differentiate for our better understanding in the initiation of translation, the elongation of translation, and the termination of translation. These three steps. Of course, in the cell, it's going on continuously. It's just for us to memorize the process better. We group into initiation, elongation, and termination. Okay. And for each of these steps, we need different factors, different translation factors. Okay. When translation starts, we have to form an initiation complex between our messenger RNA and a small ribosomal subunit. And the RNA has to bind in a way that the start codon on the messenger RNA is located at the proper site on the small subunit that is called a P site. Okay, only the start codon will go to the P site. How is it possible? because there are several actually start codons, not only the real one, but as I told you, in the sequence, you may have several AUGs. How is only the correct one recognized and put into that P site? That works by some sequence conservation around the start codon. In the bacteria, that site, that conserved base sequence is called a Shine-Delgado sequence. Shine-Delgado. These are two researchers who discovered this. They sequenced these regions of bacterial transcripts or genes and found out there is some sequence conservation. And if you make mutations in that sequence, then you will lower translation efficiency a lot. You will get less transcripts. So it's important. And this Shine-Delgado sequence is only found next to the correct start codon, not other AUGs that may be somewhere in your sequence. Okay, so that is how the correct start codon is found in the bacteria. In the eukaryote, we don't have this Shine-Delgado sequence. We have also a little bit of conservation there, but not this kind of sequence. And another thing that helps also is the cap. The cap also helps to bind to the ribosome, to the small subunit. Okay, if you would remove the cap, it would be have dramatic effects on your translation efficiency. Okay, let's go on here. So the amino acid is added to the tRNA. Okay, you can see we have the 3' base here, which I told you is the A, the CCA, and the A is the last one, the adenine. And you can see that then the amino acid here is added at that point, and we have the pairing between anticodon on the tRNA and codon on the messenger RNA. And now they try to show schematically this amino acyl tRNA synthetase, enzymes here, and have a blue side where the amino acid can bind, a black side down here where the tRNA will bind, and another one for the energy, ATP. These enzymes need ATP to get energy to connect the amino acid to the tRNA. Okay, so they form what they call an amino acyl AMP complex here, and then the transfer RNA binds to it, and that what they say, the tRNA is charged with the amino acid. If the enzyme this enzyme makes a mistake, it can still correct it at that point. Okay, as long as this complex is there, it may be still corrected. If that complex tRNA and amino acid leaves the enzyme, then it's too late. Okay, normally the process will work very fast when it's correct, and it will work very slow, the connection, if there is a mistake between a mismatch between amino acid and tRNA. Okay, so what they say, if it's the correct thing, they will assemble quickly and dissociate slowly. If it's the false one, it will associate slowly and dissociate rapidly from that enzyme. Okay? And if it's wrong, then it can be hydrolyzed as long as it's at that enzyme still bound. But if left the enzyme, then you cannot correct it any longer. You can make an experiment. Researchers actually always try to confirm what's going on. So they make by chemical modification, they changed the loaded tRNA that had normally the cysteine on it by some chemical. They remove the sulfur, which will change this amino acid into an alanine. And then they had a look what kind of peptide they get later on. Will they find the cysteine or will they find the alanine? And they found the alanine in the growing peptide later on. And so it means there is no correction any longer. Okay, the tRNA that has been loaded with the amino acid will just put in this amino acid. Okay, it will not be corrected at the ribosome. The ribosome has no mechanism to see that is a false amino acid there. Okay, it will just take what comes in with the tRNA. Okay, the ribosomes look a bit different between bacteria and us. There are subunits, large and small subunit, what they call 50S and 30S in the bacteria and 60S and 40S in the mammals. S, I don't know if you know that, is the unit for Svedberg. Svedberg unit, okay, is a sedimentation unit in the centrifugation. Okay. And then you can see the ribosomal RNAs also have a bit a different size. So in the bacteria, we have as the largest one 23S, the next one is 16S, and then we have a 5S as the smallest. And you see the number of bases in these RNAs. In the mammals, in our ribosome, we have 28S as the largest RNA, followed by the 18S, and then we have two more, 5.8S and 5S. Okay. And the number of proteins that are in the two subunits is also different between the bacteria and the mammals. So we have more proteins than the bacteria. Okay. If you make a gel electrophoresis, if you isolate RNA from an organism and you run in the gel, the agarose gel, and separate, then you can see your RNAs degrade or not when you follow these ribosomal RNAs, because as I have shown to you, they are very abundant and they form bands, whereas the messenger RNA is a small amount and you don't normally see bands in your gel. So it means you have to see this ribosomal RNA bands in your gel. If you see them sharply, it means your RNA is okay. If they look fuzzy or are gone, that means your RNA is degraded. So if you look ever for the quality of RNA, you have to look for these ribosomal RNA bands. This is a two-dimensional drawing of 16S from the bacteria. Of course, it will fold up a three-dimensional structure that looks more complicated than this. But they indicate here with this red lettering positions where they found when you make mutations what the effect is on translation. So there are many positions where you have mutation that you get a problem in the translation. Okay, termination suppression and needed for P, T, and R tRNA binding. Also another one, termination suppression, subunit association, and complementary to messenger RNA, accuracy. So there are many important areas in the RNAs. They are really functional, important for the ribosome. They don't give only a structure to the ribosome together with the protein. They're important for the function. Okay. So under the electron microscope, you are able to see them, the ribosome as the subunits and as the complete unit. Okay. And if you compare the size of the ribosome to the tRNAs and the messenger RNA, they look approximately like that. They use the angstrom as a unit. Normally, we use a metric system. So how is that? Angstrom to nanometer, do you know? 10, 10%, yeah. Okay. So the ribosome can bind in its length around 35 bases of the messenger RNA. And it can bind two RNAs at the same time. Okay. If you want to add a third tRNA, one of the first two tRNAs has to go away. There are several sites in a ribosome. So if you have a cross section, we have our messenger RNA here in white. We have a specific site where the messenger RNA is bound on the small subunit where certain proteins that are numbered here are important to bind it. And then we have three sites where tRNAs can bind: the A site or amino acyl site, the P site or peptidyl site, and the E site or exit site. Okay. The first amino acid brought in should be for the methionine, because we have the AUG, the start codon. But it's brought in by a very specific tRNA that will bind only a modified methionine that will be at the beginning of the protein. And this tRNA will go to the P site, whereas all the following tRNAs will first go to the A site. Only the tRNA that brings in the starting methionine for the start codon will go to the P site. All others go to the A site first. So tRNA and messenger RNA move through the ribosome in the same direction, I show. Huh? So the normal way, then it would be A side, P side, E side, and away. So we have the initiation, elongation, and termination steps. In the initiation, we have first that our messenger RNA has to bind in the correct way to the small subunit so that the start codon is in the correct position. And then the large subunit can come. Okay. In the elongation, the ribosome moves along the messenger RNA. Okay. And the tRNAs come in first in the A site, and then you get the peptide bond. And then the one that is in the P side will go to the E side, and the one that is in the A side will then move to the P side, and so on. And in the termination, when we have the stop codon, there is no matching tRNA that can bind. But we have a termination factor that looks in its structure similar to a tRNA, and that can bind in that place and signal termination. Then at that point, the peptide chain will be released, and the small and large subunit will dissociate from each other. Okay, and we reuse everything in this cycle between initiation, elongation, and termination. All the factors are reused. It's not like a ribosome is used one time and then is degraded. That's not the case. That wouldn't need too much energy. So they're recycled. Okay. After termination, when they separate into the small and large subunit, they can be used again for another translation step. The same for the initiation factors, the elongation factors, and termination factors. They are not thrown away after one use. Okay, they're recycled. It costs enough energy anyway, and if you throw away everything after one time, it would be too expensive. Would not be achieved at all. Okay, so initiation means we need the initiation factor. Okay, without initiation factor, nothing will happen. Okay. And the initiation factor has to go away before the large subunit of the ribosome can then bind. Okay. I told you we have a very specific tRNA and methionine for the start codon. The methionine contains a formyl group. Okay. And that is only used for the methionine at the start codon, not for internal AUGs. Okay. And this complex will only bind to the P side of the ribosome. Okay. So only this special combination of tRNA and formulated methionine will go to the P side partial P site, as I say, because we need the larger subunit to get the complete P site, and bind there. Okay, and all the other tRNAs with the amino acid go into the A site. So this tRNA is a unique tRNA. It can be used only to carry this formulated methionine. Okay, it's only for this specific case. That's why it can also go directly to the P site, because it has a special structure. Okay. We need other factors, initiation factors, okay, and we need energy, GTP in this case, okay, and we have to, after binding then of this tRNA, cut off then one phosphate from our GTP to get GDP, and then these initiation factors are released, can be reused in another cycle, and we get the complete ribosome. You may not find that formulated methionine in the mature protein later on. Either an enzyme will take that away, the formulation, or the whole amino acid is cut away. So it's modified your protein. Okay, if of course researchers try to look for that, what happens, and they said in most cases it's gone, it's not there any longer. It's only used to initiate a translation later on in the protein. You don't find that any longer. Okay, now this is the discovery of the Chantal-Delgado sequence. And so what the researchers did, they collected ribosomes with the bound messenger RNA, and then they used nuclease to cut away all the free RNA not protected by the ribosome. So they are left only then after that digestion, after they remove the protein, with that piece of RNA that was protected. And then they sequenced this. And they noticed that upstream of the start codon, they have a conserved sequence that is nowadays known after these guys who found it, Chantal-Delgado sequence. Okay. Bacteria. Okay, that is the case in bacteria. Eukaryote, we don't have this Chantal-Delgado sequence. Bacteria, I told you, they can translate at the same time their structural genes that are on one transcript. So it means the ribosome can independently bind at the coding region, initiate. So that would be the first structural gene with the first coding region on the transcript, and that would be the coding region of the second structural gene. And it will have independent translation. Okay. And it might be that further up still the RNA polymerase is working to finish the transcript. That's all what's going on in the bacteria at the same time. How about in the eukaryote? In the eukaryote, we need the cap, and we also have some ribosome binding site close to our start codon, but it has nothing to do with the Chantal-Delgado sequence. Okay. So the small subunit binds to the methylated cap first. That's why I told you if you remove the cap from the transcript, from the messenger RNA, you get a very bad translation because the efficiency to bind to the ribosome is very bad in that case. Okay, so that means first it binds via the cap, and then it scans along our messenger RNA until it finds this ribosome binding site with the start codon next to it. Okay, and then the large subunit can come in. If our transcripts are long, then at the same time several ribosomes can bind to one transcript in the eukaryote. Okay, if you have a very short transcript, then maybe only one ribosome can bind. But if there's enough space, several ribosomes can bind, and it means at the same time you will get several proteins synthesized, but the same kind of protein. Okay. In the eukaryote, they also need initiation factors. They have an E in front of it to indicate it's the eukaryotic initiation factor. They also need the GTP as the energy, okay, and then similar to the prokaryote that it goes on. And in the whole process, there are more. You don't have to remember all the names. That is unimportant for you. You just remember there are quite a number of elongation initiation factors here, more than in the bacteria, that all required that RNA, messenger RNA, and the proper tRNA with the methionine can bind and form the initiation complex, and that we need the GTP to get energy, and that then at the end these initiation factors go away and the large ribosomal subunit will come and we get the complete ribosome. So then in the elongation step, we need other factors, now elongation factors there, and we need again energy in form of GTP, where we cut off again one phosphate group, and then the amino acids now come into the A site, okay, and then the peptide bond will be created between this amino acid and the peptide that sits in the P site, and so on and so on. And this is all these factors will be recycled, never thrown away, will be recycled. Okay. So we have kind of enzyme activity in the ribosome that actually also depends on the RNA that forms this peptide bond between these amino acids. Okay. And we get the growing peptide chain that is then transferred to the amino acid in the A side first, and then the one in the P side. It will move on to the E side and go away. And the one in the A side will move on to the P side. That is actually happening because the ribosome moves along. Okay. Translocation. Okay, what happens then at the termination? At the termination, we need so-called release factors or termination factors that have a similar structure to other factors. Elongation factors G, for example, has a similar structure as the elongation factor TU together with the loaded tRNA. So they all can bind to this A site. Okay. And when we have a stop codon there, then these release factors will bind there and signal the end of translation. This is just a molecular model of a tRNA on the left, loaded with the amino acid and some elongation factors shown here in red, green, and blue. Okay, and this part is the tRNA, and that here is the elongation factor G, which is only protein. And you can see this part of the protein looks very similar in the structure to the tRNA. That's why they can both bind to this A site. Okay, I told you we may have several ribosomes at the same time on larger transcripts. Under the electron microscope, you can see that, and it's called a polyribosome. Polyribosome you get then in the eukaryote produced several copies of your protein at the same time. That is how it looks under the electron microscope. And at the end, in the bacteria, because everything is in parallel, we have our transcription and translation in parallel there, and everything is degraded actually. So that means our transcript may be used only one time. So you have translation only one time, and then the transcript is already degraded. In the eukaryote, the transcript, as I told you already yesterday, can be stable not only for hours but days or even weeks. Okay. In the eukaryote, the protein that was produced on the free ribosomes, it may go to some other part in the cell based on certain signal sequences that it carries. May go to the mitochondria, the chloroplast nucleus, or peroxisome, and you can see they have signal sequence. It means certain combinations of amino acids that act as a signal to transport this kind of protein to certain parts in the cell. If the ribosomes are bound at the membrane of the ER, we have this RER, rough ER. In that places, we have translation of proteins that go into the ER and then are transported via Golgi somewhere else. Okay. They go into the ER because they contain a so-called signal peptide at the N-terminus, a signal peptide that allows them to penetrate through the membrane, which means the signal peptide is not allowed to carry a charge because the lipid would not like to have charged molecules go through it. Okay, and this signal peptide is later on cut off. Okay, the secondary modification. And then depending what other modification you will get or what other signals are present in your protein, it will be transported to different places again. So that means in the eukaryotic cell, we have quite different things going on compared to what happened in the bacterium. They don't have these organelles there, they have no nucleus, they have no Golgi, and so on. And so the eukaryotic cell allows much more complicated ordering and transport of protein in specific compartments than what is possible in the bacteria. Okay, so my hours are finished for today. That was my last lecture. Now, I think I could have stopped this one here before, and